Good morning. My name is Stacy Kim from the University of Tsukuba in Japan. I'm currently working as a postdoctoral researcher at the IIIS, which is International Institute for Integrative Sleep Medicine. Today, I'll be talking about our most recent paper published in Nature titled Kinase Signaling in Excitatory Neurons Regulate Sleep Quantity and Depth. So sleep is a universal behavior observed in almost all animal species, from the nematodes to the higher order mammals. There has been increasing emphasis on well-maintained sleep, as the lack of and/or disturbances in sleep can have a detrimental effect on overall health of an individual. The mammalian sleep transitions cyclically from wake non-REM to REM transition, and it is a quantifiable behavior. Each vigilant state is defined by the characteristic brain activity measured by the electroencephalogram and the muscle tone measured by the electromyogram. The wake state exhibits a low amplitude and fast EEG with active and variable EMG. The non-REM sleep, or non-rapid eye movement sleep, exhibits high amplitude by delta frequency dominant with low EMG tonus, and the REM sleep, the rapid eye movement sleep, is characteristic of the EMG atonia with the low amplitude EEG dominated by the theta frequency. So the sleep-wake cycle is an intricate process controlled by a complex cascade of factors. The two-process model theory stipulates two major factors, the homeostatic process S and the circadian process S, that govern the sleep regulation. So we know much more about process S, process C, the circadian rhythm, which is controlled by this oscillatory transcriptional of the effector genes that are affected by mostly the sunlight, and modulates a vast different physiological phenomenon in our body. Whereas the process S, which is driven by the changes in the sleep need, is less known, and its molecular factors, the regulators, are less known. And Alexander Boubet has beautifully simulated this change in delta dynamics or process S dynamics, that after the sleep deprivation, there's a steep increase in the slow wave activity, or the delta power, during the non-REM. This can be simulated mathematically. So to tackle this mystery in sleep and regulation, our lab employed the four genetics study using mice. We used a large-scale EEG, EMG-based screening, dominant screening of sleep and wake behavior in mice. In this screening, the F1 founder is it's a offspring from the ENU mutagenized male. And once we are screened and identify the phenodeviants, they're back-crossed with the counter strain B6N female to produce the NG progeny. And from the NG progeny, at least 20% of the male exhibits where they exhibit the inherited behavior. We further analyze and identify the candidate genes by QTL analysis and whole exome sequencing. And in 2016, our team reported a sleepy mutation or sleepy allele in SIK3 genes that led to an increase in non-REM sleep time and the delta power during the non-REM sleep. So this sleepy mutation is a gain-of-function mutation in splicing donor site of the exon 13 and in the mutant transcript of exon 13, which is also a PKA phosphorylation recognition site, is deleted. So we generated a SIK3 flux mice to induce a conditional knockdown in the brain-specific regions. And by crossing with the nesting Cree mice, we were able to confirm that this fluxed line can induce this specific conditional knockout. And when it was SIK3 loss was induced in the glutamatergic positive VGLUT2 positive glutamatergic neurons, that we saw the opposite trend in the non-REM sleep time with a decrease the delta power, whereas compared to the gain of function mutation in sleepy. So then we continued on with our screening, and in an independent screening study, we identified a second long sleeper mutant pedigree called sleepy2 and retrospectively genotype this gene, you can see the mutant carried show shortened total wake time over 24-hour period compared to the wild type or non-mutant carriers. The LOD and QT analysis identify the chromosome 1 to be the where the candidate gene is located, and the haplotype analysis also showed this long sleepers carry this JN SNP in this particular region. So we performed the whole exome sequencing and identified that 10 out of 12 long sleepers or short wake time mice with the short wake time had the single nucleotide change mutation at the splice site of the gene HDAC4. And by direct sequencing, we confirmed that at the splicing acceptor site of exon 23 in HDAC4 had an A to T or T2A change. And HDAC4 belongs to the class 2A histone deacetylase family, and there are 4 members, HDAC4, 5, and 7, 9, and they share highly conserved deacetylase or catalyst domain. However, in mammals, the deacetylase activity is almost minimal due to a missense mutation. And so thus we generated by CRISPR-Cas9 technology to induce this identified single mutation in the splicing acceptor site in all 4 members of the family. And this spliced transcript results in the skipping of the subsequent exons, and which turns out to be a nonsense mutation that in the transcript and also in the translated proteins, there is a early stop codon and the C terminus is flanked. And we confirm by RT-PCR that these mRNA transcripts show decreased expression in these splicing isoforms. So we screened for their sleep phenotype in all 4 SA or splicing acceptor site mutant mice. And interestingly, only HDAC4 SA mutant, which mimics the sleepy2 mutant allele, and HDAC5 splicing of the mutants showed decreased wake time, whereas HDAC7 and HDAC9 does not show any changes in sleep and wake. So in this HDAC4 and HDAC4 splicing acceptor mutants, the decrease in the wake time was compensated by increase in total sleep time, specifically more prominent in the non-REM sleep, and also showed increase in delta power during the non-REM sleep. And in HDAC4, as we saw in the RT-PCR results, these splicing mutants are loss of function type, and HDAC4 systematic knockout mice are known, homozygous mutants are known to be perinatal lethal. So we were not able to screen or observe sleep phenotype in the homozygous mutants, whereas the HDAC5 homozygous mutants were also viable and relatively healthy. So we were able to record their sleep phenotype, and those dependently that they showed increase in the non-REM sleep time as well as delta power. So since HDAC4 and 5 are known primary target of SIK3, we wanted to assess the interaction between SIK3 and HDAC4 in sleep and wake control. SIK3 has been known to have very critical 2 phosphorylation sites, the T221 residue, which is critical for its kinase activity, and S551, which is a PK recognition site that lies within the exon 13 that will be deleted in the sleepy allele. And HDAC4 and 5 have 3 conserved phosphorylation sites that are known to be critical in their transcriptional repressor activity. And the mode of mechanism for HDAC4 and 5 is well established in nucleocytoplasmic shuttling that is mediated by the phosphorylation of these 3 sites. So we assessed first if there was any change in the HDAC4 localization based on the change in sleep need or the time of the day. So at ZT6 and ZT12.5, ZT6 will be when the mice are sleeping, thus the sleep need is low or decreasing. And at ZT 12.5, just after onset of dark, so this will be the lowest point of the sleep need, thus these mice will be awaking. And we've confirmed that in the motor cortex and somatosensory cortex, that the nuclear HDAC4 intensities were increased, they're greater at the ZT 12.5, where the sleep need is lower and mice are awaking. And at the same time, ZT 12.5 in sleepy mutant mice, the nuclear HDAC4 intensity was lower, which is expected since sleepy, the gain of function mutant, increases phosphorylation at HDAC4, which will be touched in the later slide. So we performed the cytoplasmic nuclear fractionation from using the mouse cortical tissues after sleep deprivation or at the ad libitum sleep, wild-type mice. And we observed that the nuclear localized HDAC4 was decreased when in the sleep-deprived animals with the high sleep needs. Those changes in sleep need mediate the nuclear cytoplasmic shuttling of HDAC4. So we then tried to confirm or establish the epistasis between SIK3 and HDAC4. So in vitro kinase assay, we were able to confirm that out of the 3 phosphorylation sites, the S245 residue was a primary target for SIK3 using a different kinase recognition peptide known for SIK3. So, and this in PC12 cells, we also confirmed that using the 10 SIK3 inhibitor that the phosphorylation site level at S245 residue was decreased dose dependently as SIK3 was inhibited, whereas at S629 did not have such an effect. And also in the vivo in brain homogenates of the sleepy mutant mice, that we confirmed that there was an increase in phosphorylation at S245A residue of HDAC4. So again, in vitro study, we wanted to see the effect of sleepy or the 551A mutation mimicking the sleepy phenotype have an effect, how they affect the transcriptional repressor activity of HDAC4. So using the MEF2, a known transcriptional cofactor of HDAC4, we confirmed that when SIK3 is co-expressed, the transcriptional repressor activity of HDAC4 is diminished. And this is further enhanced by S55A mutant, which is, again, a dominant gain of function mutant type. And TT21A, which is kinase dead form of SIK3, does not have any effect on the transcriptional repressor repression. And then we wanted to confirm whether this phosphorylation site 245 is critical for this transcription repressor activity. And yes, that when we express the 245A mutant of HDAC4, the transcription repression is further enhanced. And with the HDAC4 wild type and SIK3, this de-repression was further enhanced. However, with 245A mutant, SIK3 wild type nor the S551 mutant had such an effect, thus indicating SIK3 phosphorylates S245A, the serine 245, and this phosphorylation is critical in the shuttling of the HDAC4 or its effect on transcriptional repression. So using the CRISPR-Cas9 system, we created this phosphodefective mutant mice. And in these mice, in heterozygous mutants, also show the opposite trend of both SIK3 sleepy and the HDAC4 haploinsufficient mice, that there is a decrease in non-REM sleep time, as well as a decrease in non-REM sleep delta power. And to, again, confirm the epistasis between SIK3 and HDAC4, we adopted this PHP EB serotype of AAV, which can penetrate through BBB, and it does very useful in transducing the virus in entire CNS. So we overexpressed HDAC4 245A with 5AV in the sleepy SIK3 sleepy mice. So again, the SIK3 sleepy have increased non-REM sleep time and increased delta power. And by expressing S245 mutation in sleepy mice, the hypersomnic phenotype of the sleepy mice were abolished. So you can see there is no increase in non-REM sleep time almost to the level at the level of the wild type. And the delta power also showed an increase or restoration towards the wild type level. And also the delta power shows the same trend. Thus, this confirms that SIK3 directly phosphorylates S245 of HDAC4, which is critical in the regulation of non-REM sleep time and also the delta power. Then to confirm this conserved role in SIK3, HDAC4 in sleep regulation, we've collaborated with one of our members in our institute using the C. elegans. So using two mutant worms, Kin29 is SIK3 homolog, and this particular mutant is a kinase DET mutant, and HDA, HDAC4 ortholog is the null-like mutant here. So when the kinase DET form of SIK3 is expressed in the worm, that there is a reduction in fraction of quiescence, whereas the loss of HDAC4 or HDA4 in these worms increased the quiescence. However, when you express HDA, the knockout of HDA4 in the kinase DET mutant of SIK3 worms, the phenotype of the decreased fractionation did not occur. And also confirming that in HDA4 knockout worms, when we overexpress the wild type HDA4, the increase in the quiescence was diminished and returned to almost at the basal level. So again, indicating that SIK3 HDAC4 signaling in sleep regulation is conserved in different species. So then next, we wanted to assess the importance of the involvement of the kinase activity of SIK3 in this signaling pathway. So first, one of the well-known upstream regulator of SIK3 is LKB1. LKB1 phosphorylates SIK3 at 3-221 residue, which is critical for its kinase activity. And in the in vitro assay, in two different mutants, so 221A, which is phosphodefective and thus kinase activity is diminished. And in 221E mutant, which is phosphomimicking, but is known to be independent of LKB1. And you can see the kinase activity in both mutants were some decreased. And in our 2016 Nature paper, we also reported that after sleep deprivation, the phosphorylation at T221 is increased, thus indicating that there is the importance of this kinase activity in non-REM sleep or in sleep homeostasis. So we use the pre-induced, the inducible Cree in the synapsin1 Cree-ERT line crossed with the LKB1 flux mice to make conditional knockout in neurons, in brain-specific conditional knockout of LKB1. And these mice here, then the LKB1 with the knockout that it's not able to phosphorylate T21, thus activating this SIK3 kinase activity. And these mutant mice that showed decrease in non-REM sleep time and decrease in delta power, indicating that the kinase activity or phosphorylation T21 threonine of SIK3 by LKB1 is important in the non-REM sleep regulation. Then next, we assessed this T221E, the phosphomimicking mutants. And these mice, although did not show any changes in non-REM sleep time, it did show slight increase or restoration of delta power. But slight increase in the delta power. So partially rescuing the conditional knockout phenotype of LKB1. Then in SIK3 T221A mutant mice, in same pattern that there wasn't significant changes in non-REM sleep time, but we saw a significant change or reduction in the delta power in these phosphodefective mutant mice. So to further confirm the importance of the kinase activity in the hypersomnic phenotype of SIK3, we expressed 221A mutation into the sleepy allele. And these, compared to the sleepy mutant with the hypersomnic phenotype, when we introduced the phosphodefective mutation of T221, thus the kinase activity is inhibited, there is a decrease in non-REM sleep time and decrease in delta power during non-REM sleep, indicating that kinase activity of SIK3 is critical for the regulation or changes in non-REM sleep time and delta power in these mice. So then we further wanted to investigate then where and what type of neurons and regions in the brain the signaling pathway is important. So we adapted two flux lines, actually 3 flux lines, but first the HDAC4 flux line to induce the knockout or loss of HDAC4 in specific cell mouse cell types and regions, and also the sleepy flux to induce the sleepy-like the skipping of exon 13 in pre-dependent manner. So first, in glutamatergic neurons, both in HDAC4 showed that there's an increase in non-REM sleep time and delta power during non-REM sleep when HDAC4 was lost in VGLUT2 positive glutamatergic neurons. And using the chem kinase 2 alpha Cre line, which is mostly forebrain excitatory neurons, we did not see any changes in non-REM sleep time. However, there was an increase in the delta power during non-REM. So in sleepy flux mice, again, these are the gain of function sleepy type of mutation. So in both VGLUT2 in the same, the trend was the same in VGLUT2 positive excitatory neurons. Induction of sleepy allele, sleepy increased the non-REM sleep time and delta power. Also in COM kinase 2, the forebrain excitatory neurons, induction of sleepy in the same manner did not show any changes in total sleep time, non-REM sleep time, but showed increase in the non-REM delta power. However, in VGAT, the VGAT positive GABAergic neurons, neither the loss of HDAC4 or the induction of sleepy allele showed any changes in total sleep time and delta power. So then we next used the two different region-specific Cre lines. One is FOXG1, which is the developmental marker for telencephalic lineage of neurons, and FOXD1, which is pre-hypothalamic marker for pre-hypothalamic neurons. And for when we induced SIK3 sleepy mutant allele in FOXG1, so the forebrain specific or cortex or forebrain specific induction of sleepy allele, there was no change in sleep time, but there was a significant change in terms of the delta power during the non-REM sleep. Whereas when sleepy was induced in the hypothalamic neurons only, there was a significant changes in the sleep time, non-REM sleep time. However, there was no change in delta power. This indicates that the delta power slow wave activity during non-REM sleep and the total time spent in non-REM sleep are controlled, regulated by different regions in the brain. And the interplay between these different regions is important in terms of the responses in the regulation of sleep homeostasis. So then this was again confirmed in the sleepy or SIK3 knockout mice. So when we knocked out SIK3 in fecal T2 positive glutamatergic neurons, the trend was the opposite from the sleepy or loss of HDAC4 with the decrease in the total non-REM sleep time, the decrease in delta power. And again, in the VGET Cree line, when SIK3 was knocked out, there was no change in the sleep time and delta power. Thus indicating the SIK3 and HDAC4 signaling is critical in excitatory neurons of the cortex and hypothalamus. So this is a summary of our finding. So the regulation of sleep death, or indicated by the non-REM delta power and episode duration, is controlled and regulated by the glutamatergic neurons in the forebrain or the cortex. However, the sleep quantity, the time of the non-REM sleep, is controlled by the excitatory neurons in the hypothalamus. So then we wanted to further investigate then what are the downstream factors of this pathway. So we performed single nucleus RNA sequencing using two different schemes. First was to express SIK3 wild type or SIK3 sleepy in the motor cortical regions. And the second scheme is in sleep deprived for 6 hours. Mice that were sleep deprived for 6 hours, and the control were left to sleep and wake at libidum. In both cases, the major clustering of the differentially expressed genes were glutamatergic glutamatergic, and there was a 33 upregulated genes changed both in both schemes with sleep deprivation model or in SIK3 models. And there are about 5 differentially expressed genes between the sleep deprivation model and the sleepy model. So this is a little volcano plot. So many of the genes identified in both schemes, both models, were biased towards the SNAPs-related or synaptic plasticity-related genes, such as BDNF, which has been reported to be critical in sleep homeostasis. So this is a little summary. So in the wild type or in general settings, the LKB1 phosphorylates SIK3 and induces its kinase, activates its kinase activity, whereas PKA phosphorylates S551 residue, which may be inhibitory in its downstream effector. And this activated SIK3 then in turn phosphorylates HDAC4 and 5, and this phosphorylation of HDAC4, 5 sequesters them into the cytoplasm, and their repressor activity in the nucleus is relieved as the transcription of sleep-promoting genes are enhanced. When sleepy, however, is expressed, these are gain-of-function or hyperactive form of SIK3, and this phosphorylation level is increased in HDAC4 and 5, thus further sequesters them into the cytoplasm. Then the sleep-promoting gene expression is no longer homeostatically controlled by the cytoplasmic and nuclear shuttling of these HDAC4 and 5. So in sleepy mutant mice, that the induced sleep or increased sleep is observed. In HDAC4SA with the haploinsufficient model, that the total expression of HDAC4 is decreased at basal. Thus, the repressor activity is also reduced and promoting the transcription of sleep-promoting genes. And I would like to thank all the members of our lab. This project was truly a collaborative effort, as you may have seen, that many of us got together to put this great paper together. And I really thank everybody who was involved. So this is a nice summary of our graphical depiction of our study. So LKB1, SIK3, HDAC4 signaling is important in this sleep and wake regulation, especially the non-REM sleep time and death regulation. And this occurs in the excitatory neurons in the animal, and the sleep death is mainly regulated by the cerebral cortex, whereas the sleep quantity is regulated by hypothalamus. And this is the end of my presentation. I think I spoke too fast, but if you have any question, you're more than welcome to email me and I can take questions now.